guys! Welcome again to another video of Pag-aaral, and in today's lesson, we're going to discuss meiosis and the sexual life cycle. So these are the things that you must know and you must learn at the end of this video, and basically we're just going to discuss these things. So one is the types of reproduction, next is meiosis, the steps during the process of meiosis, third is the sources of genetic variation, and fourth is the difference between your mitosis and meiosis. So as an introduction, since we're dealing with reproduction, with sexual cycle, so reproduction, through the process of reproduction, offspring acquire genes from parents by inheriting chromosomes, and we define genes as the segments of DNA that code for basic unity of heredity. Now, as you can see here, dito sa family picture namin, we can say na we inherited this characteristic from my mother, yung parang, yung gantong cheeks. So, kita niyo naman sa mama ko. So, that's an example of how the gene is passed from one generation to another, from parent to the offspring. Now, what are the types of reproduction? So, we just have two, asexual and sexual reproduction. So, asexual reproduction often produces clones, and it just requires single parent. That's why there's a little variation kasi wala namang another parent to contribute the gene. So, isa lang yung parent niya, and because of that, asexual reproduction is much more faster and energy efficient. So, example of this are budding and binary fission. So for sexual reproduction naman, basically it is observed in multicellular organism. So one type of sexual reproduction, or parang one process under it, is the meiosis that produces gametes. So it is from two parents, so female and male. So it has a lot of variation, thus it has a lot of diversity. So it is lower compared to asexual reproduction. And example, or the organism that has the sexual reproduction is humans. And another example din pala yung trees. So what are the varieties of sexual life cycle? So we have like different varieties depending on the organism. Like for example, if we have an animal, it follows this kind of life cycle. And then here we have the plants and some algae. So they basically follows the alteration of generation. And next we have this for the fungi and some protists. And also we have this human life cycle. Well, we know that when the egg is fertilized by the sperm, and fertilization happens, it will develop into a diploid zygote, and that diploid zygote will undergo several mitosis and development until it reaches this multicellular diploid adult, and then this adult, through their ovaries or testes, that organ will be able to produce or undergo meiosis to produce the haploid gametes that later on will come into contact again to develop a diploid cycle. So that's how the human life cycle occurs. Okay, so just a review, we have here your chromosome, and then your chromosome is composed of the P arm, and then P arm here, and then you also have the centromere and the sister chromatid. So the centromere is the one that attaches your sister chromatids together, and the Q arm, of course, which is the long arm. So in chromosome, specifically, in somatic cell, we have 46 chromosomes, and 22 pairs of it constitute to the autosomes, and then the last pair is the sex chromosome, so that would be X and Y. So for females, we have XX, and male, it has XY. So for gametes, we just have 22 autosomes plus 1 sex chromosome. Now, these are the words to remember, or parang you need to be acquainted with, especially here dito, especially in our topic especially our topic is about meiosis. First is synapses. So synapses is just the pairing of the homologous chromosome forming a tetrad, and we define tetrad as two chromosomes or two homologous chromosome of 4 chromatids. So kaya siya tetrad kasi it's apat. And then when synapses happens, there's also going to be a crossing over or the exchange of genetic material, and the site of the crossing over we call that your chiasmata. And these homologous chromosomes naman, so these homologous chromosomes are basically a pair of chromosomes coming from maternal and coming from paternal that are similar in shape and size. So homologous pairs, so tetrads carry genes controlling the same inherited trait. So for example, here this blue one came from the paternal and then this red one came from the maternal. So they will be paired together, may mangyayaring synapses because they are considered to be homologous chromosomes. So not only they are the same when it comes to their shape and their size, they also contain the same gene or the same gene controlling the same inherited traits. For example, ditong part na to, nandyan yung gene for the eye color. So ito din is meron din siyang gene for the eye color. So that's why they are homologous. So hindi naman necessarily na dapat parehas sila nung type of gene na yun. For example, dito yung gene color niya is red, is blue. Tapos dito is blue din, hindi naman. So kahit magkaiba, depende din syempre kung ano yung characteristic ng parent. Okay? So why are homologous chromosomes not exactly identical replicas? Kasi nga, they are just homologous, not replicated ones. Like, katulad dun sa sister chromatids, diba? Una, one strand lang siya ng chromatid, and then it undergoes replication. Now you have sister chromatid. Pero here kasi, one pair of the chromosome came from the paternal and one from the maternal. That's why they are really not the same. Okay, so karyotype. Karyotype is a picture of an organism's complete set of chromosomes that is arranged from largest to smallest pair. So this is an example of a karyotype. So as you can see, one pair came from maternal and then one pair came from paternal. So from this example, what can you say? Is it a male or female? It's a female kasi dalawa yung X mo. Okay, so now let's go to your meiosis. So meiosis basically involves two successive divisions of a diploid nucleus. So first is, we call that your meiosis 1, and second is your meiosis 2. Okay, so what happens during meiosis 1 is basically the separation of the homologous chromosomes. Diba? Kasi we discussed kanina na your homologous chromosomes, they need to be paired with one another, tapos they need to be divided. So, so that basically happens during your meiosis 1. So, in meiosis 1, we have these 4 phases. So, prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay, so before we start with the prophase 1 pala in meiosis 1, let's just define or parang discuss what happens during interphase 1. So basically what happens there is just the same of what happens during the mitosis interphase. So similar, to the mitosis, your chromosome starts to replicate then in the S phase, and each duplicated chromosome consists of 2 identical sister chromatids attached at their centromere, and the nucleus and nucleolus are still visible, just like what is observed in the mitosis. So what happens during your prophase 1? So what happens during your prophase 1 is katulad lang din ng sa mitosis basically. Pero we're not going to concentrate on that kasi you already know that. So, concentrate tayo sa homologous chromosome, alright? So, when I say na parang the same lang, ibig sabihin your centrosome will move at the opposite poles and then mag-disintegrate na yung nuclear envelope nyo to make way for the alignment of the homologous chromosome. So, other than that, we also have this stages here and the parang events that sinabi ko na kanina. So, in prophase 1, may nangyayaring synapses or the homologous chromosomes pair up, and then we're going to have, of course, then the formation of the tetrad or the 4 sister chromatids, and of course, we're since merong synapses, there would also be a crossing over at the chiasmata, or the site of the exchange of genetic material. So we have here your leptotin. So what happens during the leptotin is the chromosome starts to condense. So after it condenses, ang mangyayari is it will undergo the zygotin. So zygotin, it is where the synapses begins, or the synaptonemal complex form, or the parang close pairing or interaction of your homologous chromosome. Next is the pachytene. So here in the pachytene, it is where specifically crossing over happens. So there's an exchange of genetic materials by non-sister chromatids and diplotin. So this is where the synapses end. So the chiasma visible within the pipeline. So magkakaroon ka na ng chiasma or ng chiasmata. Ibig sabihin, nagkaroon na ng exchange of genetic material and the synapses ends. So they are not really close na. Hindi yan sila super close. So they are just attached within doon sa dulo or doon sa chiasmata. As you can see here sa diakinesis. So sa diakinesis naman, the prophase ends and nuclear membrane disintegrates. So totally, dito na nawawala talaga yung nuclear envelope or the membrane. And what happens there after is the homologous chromosomes will line up at the middle to make way for the separation of the homologous chromosome. So that happens during your metaphase 1. So in metaphase 2 naman, what happens is the homologous chromosome separate, and it is not the same with the mitosis, the anaphase of mitosis, kasi sa anaphase of mitosis, what happens is the sister chromatid separates. Pero dito kasi, the homologous chromosomes separate. So they are completely different. Iba kasi pag sinabi mo yung sister chromatid yung nag-separate, and then yung homologous chromosome yung nag-separate. So please take note of that. And after the homologous chromosome are nandun na sila at parang both poles or parang different poles of the cell, the cleavage will now be developed and will undergo cytokinesis, or the entire separation of the cytoplasm. Okay, in meiosis 2 naman, this is the parang mitosis. Kasi one cell, di ba kanina nakapag-produce na tayo ng two daughter cell. So that two daughter cell will undergo pa here, one entire division and then another entire division, parang leading to the creation of four daughter cells. So what happens during prophase 2 is just the same, kasi nga during the telophase 1, di ba, parang ang nangyari is katulad nung sinabi natin before, nagpo-form uli yung nuclear envelope nyo. Pero here sa prophase 2, mawawala uli siya kasi nga kailangan na mag-line up ng chromosomes at the middle part. So here sa metaphase 2, magla-line up na yung chromosome or yung sister chromatid. Take note that it has the parang genetic material na, the exchange genetic material. So meron na tayo ditong recombinant part. So sa may chromosomes you here, and then after which it will be separated by the microtubules again, or the spindle fiber. So your sister chromatids na, they are separated from one another, and then it will undergo another telophase 2 and cytokinesis. So after the course, the cytokinesis, after the cytokinesis, it will undergo again process of meiosis if kailangan ng katawan. Pero that's basically it. That's your meiosis. So what are the events unique to meiosis 1 that is not in mitosis? So first is the prophase 1, where the synapses and crossing over happen, and then the metaphase 1, which the pairs of homologous chromosomes line up on the metaphase plate. Kasi remember, in the metaphase of mitosis, ang nagla-line up there is the sister chromatids, not the homologous chromosome. And lastly, the anaphase 1. So the anaphase 1, homologous pairs separate, and in metaphase, in mitosis kasi, ang nagsiseparate there is the sister chromatid, or the sister chromatid. In anaphase 1 then, sister chromatids are still attached at the centromere. So what are the sources of genetic variation? So since meiosis results in random separation of chromosomes and gametes, it causes diverse population that over time can be stronger for survival. Charot. Okay, so these are the sources for genetic variation. First is the crossing over or the exchange of genetic material. So what do you think will happen if nagkaroon ng genetic material or the exchange of genetic material? We will have a recombinant chromosome. So when we say recombinant chromosome, ibig sabihin, nagkaroon ng unique combination of genes or nagkaroon ng exchange of genetic material, kaya nagkaroon ka ng unique combination of genes. So we call that your recombinant chromosome, and because of that, mas kakaroon nga tayo ng higher diversity. Next is independent assortment of chromosomes. So when we say independent assortment of chromosome, it basically just parang explains or parang means that there's a random orientation of homologous pairs in metaphase 1. So here, kasi diba, for example, this came from the paternal and then this red came from the maternal. So there's a possibility that it will line up like that. Na yung chromosomes nyo, it will line up like that. But there's also a possibility na ganito yung itsura niya. Like this blue is here, and then the red, and then the red, and then the blue. So ganun yung possibility. And according to scientists, we actually have 8 million possibilities in lining up those 23 pairs of homologous chromosome. Kaya there's a lot of variation talaga na pwede mangyari in combination. So, kaya nagkakaroon din, yan, ulit-ulit tayo ng genetic variation. So, third is random fertilization. Of course, any sperm plus any egg. So, that's 8 million times 8 million. You have 64 trillion combinations. So, guys, you're unique. So, that's how unique you are. Okay, now let's differentiate mitosis versus meiosis. So mitosis happens in somatic cells. It undergoes one division lang. It results in 2 diploid daughter cells. So basically just clones of the parents and from zygote to death. So it happens during parang when you are formed as a zygote until you reach maturity or you develop along the way. And then purpose basically is for growth and repair, and no synapses, no crossing over, no recombinant DNA here. So for meiosis naman, it happens in your gametes lang, 2 divisions, 4 haploid daughter cells, genetically different, less than 1 in 8 million alike, and females before birth, follicles are formed, mature ovary release, beginning puberty, and purpose, reproduction. So it just means here, sabi dito kasi, from zygote to death, and meiosis only happens especially in female. So, ang nangyayari is before birth, follicles are formed na. So, mature ovary release, beginning puberty. So, nagpo-form na yung parang egg cells even before the actual birth, and then it will just be released into a mature ova, beginning to puberty. Okay? The purpose of meiosis basically is for reproduction. Yay! So, we have this pala, the nondisjunction. So, nondisjunction is the failure of chromosome or of homologous chromosomes or sister chromatids to separate during cell division. So, what happens when nondisjunction, what happens when the homologous chromosome or sister chromatids fail to separate during the division? Magkakaroon ka ng abnormal number of chromosome, and we call that aneuploidy. So, the nondisjunction happens in 3 forms. First is the failure of the separation of your homologous chromosome, and then failure of the separation of your sister chromatids, and third is the failure of sister chromatids to separate during mitosis. So this, for example, here, nagkaroon ka ng nondisjunction of homologous chromosome. So, ang nangyari dahil nagkaroon ng nondisjunction of homologous chromosome in meiosis 1. So, nung nag-separate sila, diba, meiosis 1 and then meiosis 2, ito na lang yung natirang chromosome. Ito, marami pa. So, pag nag-divide ulit siya, ito tatlo, ito naman kulang ng isa, kasi nga nandito sila. So, that's what happened if nagkaroon ng nondisjunction. So, same here sa sister chromatid. So, pag here, for example, sa meiosis 2, hindi nakapag-separate ng maayos yung sister chromatid. Ang mangyayari is one cell will receive an additional, and then yung isa kulang ng isa. And when non-disjunction occurs during meiosis, it can result in gametes with the wrong number of chromosomes. If fertilized, the most common result is miscarriage, and most human embryos cannot survive with an abnormal number of chromosomes. Kasi it could mean na, syempre, kulang na siya ng specific gene na kailangan ng isang individual to develop. We also have chromosomal arrangement. So in some cases, siguro hindi okay yung pagkaka-separate, na-separate yung homologous chromosome versus yung chromatin. Pero in some cases naman na nangyayari, there's a chromosomal rearrangement. So there's a large chunk of chromosome, but not the entire chromosome that are affected. So such changes are called chromosome rearrangement or chromosomal mutation. So the chromosomes are basically rearranged, and we call that aberrant chromosome. And the rearrangement often happen when crossing over doesn't work properly. So pag hindi nangyari ng tama yung crossing over, nagkakaroon ka ng rearrangement ng chromosome. So it has four types. First is the duplication, where a chromosome ends up with two or more copies of gene segments. So basically, dapat isa lang yung gene segment na yun, but nagkaroon nga ng duplication. So, nangyari is na doble. Just like here. So, dapat i-duplicate niya lang to, pero nangyari is, ayan na yun nangyari. Nagkaroon ka ng dalawang segments nito. Na times 2 siya, kaya medyo humaba dito. And then next is the deletion. So, a chromosome ends up with no copies of a particular gene segment. So, nagkulang siya ng isa. So, as you can see here, di ba, A, B, C, yung segment ng genes, pero here, meron ka na lang B, C. So, na-delete. Next is inversion. Ang chromosomal region is flipped around so that it points in the opposite or wrong direction. So, inversion here, as you can see, na nagkabaliktad silang dalawa dito. And next is translocation. So, a piece of one chromosome is attached to another non-homologous chromosome. So, parang instead of it doon dapat sa homologous chromosome sila yung magkakaroon ng crossing over, ang nangyari is nag-crossover siya doon sa non-homologous chromosome sa hindi niya kaparehas na nako-contain na gene. And yeah, that's all for your meiosis. If you have any question, just send me a message, and I hope you learned a thing or two. And guys, good luck on your exam about meiosis. Bye!